Good morning, I'm Mark Reithmer. I have the privilege of being the CEO of the Alzheimer's Drug Discovery Foundation. Welcome to our symposium at the 8th Annual Great Ladies Luncheon. I'm up here because we have a simple change in the agenda. Any of you who's been here to the symposium for the last couple of years, and you will know from wonderful to have you all back, and welcome to folks who are new to the symposium. Those of you who've been here, you know we typically have from NBC, Andrea Mitchell, who serves as the MC for the symposium. As any of us who've seen the news of the last 24 hours with the passing of Barbara Bush, Andrea needed to go to Texas last night. But we are very fortunate. Our luncheon MC is here early, and he offered to kick off the panel today. And you will notice he brought some cameras with him from CNN. So again, he's going to host this morning, he's going to host at lunch. Somebody actually does, particularly in this town, actually anywhere, doesn't really need an introduction, but he deserves an introduction. I suspect that you know him as a noted author. I suspect you know him as a wonderful debate moderator, and I know you know him as CNN's lead political anchor and host of the Situation Room. Ladies and gentlemen, I give you Wolf Blitzer.
Gentlemen out there. Thanks so much for coming. This is really, really an important day for all of us, and we're going to learn a lot right now. We have X amount of knowledge in our heads as we start this symposium right now, but in a little while we'll have X plus. We'll all be a little bit smarter thanks to these wonderful people, and I'm grateful to all of you for showing up, for listening and participating in this. It's really my pleasure to welcome all of you to this morning's symposium on behalf of the Alzheimer's Drug Discovery Foundation, or as we like to call it, ADDF. Alzheimer's cases are rising right now. As the baby boomer generation ages, check it out, its risk for Alzheimer's unfortunately increases. Today, 5.7 million Americans have the disease. Almost 6 million Americans right now suffer from Alzheimer's, but that number will rise to 14 million in only a few decades unless we find a way to stop it, and we can. The good news is that the ADDF is getting close. It's the only charity right now solely focused on finding drugs to prevent and treat Alzheimer's disease, and those drugs are clearly advancing. Today we're privileged to learn about some of these potential new therapies directly from the scientists working on them. Our panelists include Dr. Fillit, and Dr. Yuko Hara from the ADDF, who are joined by Dr. Eugenia Trushina from the Mayo Clinic and Dr. Susan Catalano from Cognition Therapeutics. Dr. Trushina and Dr. Catalano are developing novel drugs to treat Alzheimer's. Both are pursuing innovative approaches to Alzheimer's, and both have received support from the ADDF. The ADDF has always funded new ideas, often a decade or more before anyone else. Science doesn't stop. We constantly learn new things about the causes of Alzheimer's. The ADDF makes sure those findings get translated into drugs to treat and prevent it. After the symposium, I invite, of course, all of you to walk just across the hall to the luncheon. This year, the ADDF is honoring longtime partner the Neuroimaging group. Curing Alzheimer's is not easy work, and partners like Neuroimaging ensure that we keep moving forward. The fashion presentation this year by Edro, and we're all looking forward to that, and they have brought a special gift for everyone, because when you think of fashion shows, you think of Wolf Blitzer. Been covering it after so many years. This benefit event was created 8 years ago by good friends Elise and Mark Lefkowitz. Let's give them a big round of applause. And we thank them for all they do for Alzheimer's research. We also thank Eli Lilly for sponsoring today's symposium and for remaining deeply committed to Alzheimer's drug research. Now it's my pleasure to introduce Dr. Fillit, the ADDF's Chief Science Officer and Founding Executive Director, who will lead today's panel. Let's give him a big round of applause.
I want to thank Wolf so much for being here today. We really appreciate it, Wolf. You know how busy you are, and we're just very grateful to you for being here today. And I also want to thank Eli Lilly as well for co-sponsoring us. Most of you are probably familiar with basic research that's going on in academic medical centers and universities all over the world. Drug research is a completely different type of science. Basic research is essentially more often than not biology. It's figuring out how cells work, how the brain works. It's gaining knowledge about diseases. But the intent of basic research is, in a functional way, not necessarily about developing a drug. The hope is that ideas that are learned from biology will lead to the science that leads to a drug. And the science that leads to a drug is much more about chemistry than it is about biology. To create that little white pill that one day all of us could take that incredibly would prevent and treat Alzheimer's disease and remove this scourge from all of our lives. That will be amazing. And so what we're here today to discuss is how that happens. What is medicinal chemistry? What is the pharmacology that goes into it? Pharmaceutical scientists, regulatory issues that lead to the development of drug, how clinical trials are done, and how difficult they are to conduct. And we have here today Dr. Trushina, who is a professor at the Mayo Clinic, who was a pharmacologist and doing biology research, and she'll tell you about that, at the Mayo, and we helped to get her into doing drug research, and I hope she'll tell us some of the challenges she's encountered in doing that, and on a very interesting target that Dr. Trushina is developing, which is really about the way that neurons in all of our cells get energy, and why energy failure in the aging brain is critical to the development of Alzheimer's disease, and how energy failure targets can be the target for developing drugs for Alzheimer's disease. And Dr. Catalano has been a drug developer and discoverer for much of her career, and she is also a scientist, but she chose a different route to go to become a bio-entrepreneur and to start a biotechnology company that is dedicated to developing drugs for Alzheimer's disease. And early on in the establishment of her company, ADDF made an investment in that company to help the company move forward. Dr. Catalano's target for drug research is about the synapse. It's about how synapses, the connections between our neurons in the brain that help us to think, that enable us to think, the trillions of synapses in our brain that enable us to think, become disconnected in the Alzheimer brain. And when these synapses become disconnected and dysfunctional, we can't think anymore. And so Dr. Catalano's her target for drug, is to preserve those synapses and protect them from injury by molecules like beta amyloid, which many of you have heard of. And our last scientist here is Dr. Yuko Hara, who's the director of ADDF's Aging and Prevention Program. And for that, what we've chosen to do and what the prevention program does is it prevents we fund a certain kind of prevention research, which is, again, quite different than biology or even chemistry. We are looking at some drugs for prevention, and we've developed a website called vitalityorg, which gives people credible, understandable, scientific information about all the things that you hear about in the newspaper, where lay people and even doctors really can't evaluate the evidence for whether or not something really works. And the best example that I can give is that recently on the cover of USA Today, there was a major story about how bread is bad for you. And this prevention research was conducted to show that the reason that we know that bread is bad for you is that 95% of all felonies are committed within 24 hours of eating bread. And so Dr. Yuko Hara is going to explain to us what it means to understand evidence, scientific evidence. So let's begin with Dr. Trushina. Dr. Trushina, Alzheimer's has been called type 3 diabetes. And diabetes, we know, is a risk factor for Alzheimer's disease. What's the connection between diabetes and your efforts to develop a drug for Alzheimer's disease?
So diabetes, and when we talk about diabetes or Alzheimer's, we're basically talking about energy. So when in type 1 and type 2 diabetes, the mechanisms of the disease are directly linked to our reduced ability to utilize glucose. And glucose is the major molecule that powers every single cell in the body, including peripheral cells, but also the brain. So type 1 and type 2 diabetes primarily affect peripheral cells. And we know that the disease strikes when glucose cannot go into the cells, but accumulate in the blood. So the high raise of glucose levels in the blood basically tells us that the cells do not have enough glucose anymore and cannot perform normal functions. So it's all on the level of energy utilization that the failure and detrimental effects of diabetes occur. Type 1 and type 2 diabetes are the factors that could promote cognitive dysfunction. It's not necessarily that everybody with diabetes will develop dementia or Alzheimer's disease, but these are risk factors. So body is connected. The brain and the body is all connected. So what we eat, what we do with our body directly affect the brain. But it's very interesting. The brain has its own problems with glucose utilization. And glucose is the major energy fuel for neurons. So neurons communicate through passing signals from one neuron to the other, connecting to each other through these synaptic connections. And it's very important to power these synapses. So when you do not have enough energy in neurons, these synaptic connections are lost, and that's directly relevant to the loss of energy. So in Alzheimer's disease, one of the early problems detected in people with mild cognitive impairment is the abnormal glucose uptake. So it's very similar to type 1 and type 2 diabetes, but it occurs in the brain. So there is similar failure in the glucose, in the energy levels that directly affect neurons. So type 3 diabetes was the term introduced by another female scientist, Suzanne de la Monte, and she is MD, PhD, so she sees patients and she looks a lot at pathology markers in the brain and in the periphery. And she came up to the conclusion that type 1 and 2 peripheral diabetes actually resembles mechanisms of what is seen with reduced energy and glucose utilization in the brain and introduced this term type 3 diabetes. It's very interesting because, again, type 1 and 2 and type 3 diabetes not necessarily go hand in hand. So, for example, type 3 diabetes could happen without any type 1 and type 2 diabetes. But what is interesting that now we started to recognize that very important interventions that could prevent type 1, type 2, and type 3 diabetes include healthy lifestyle, exercise, and healthy living, basically. So Yuko probably will talk a lot about that. But this is very interesting because we do not understand necessarily why people develop type 1, type 2, or type 3 diabetes. But we know that healthy living and active lifestyle, especially for women in the middle age, could significantly delay the onset of diabetes, including type 3 diabetes. But the interesting unifying mechanism is on the level of energy. So we all rely on energy.
So, Eugenia, you're targeting a part of the cell called the mitochondria, which is where the cell generates energy. And your drug targets the mitochondria. Mm-hmm. So could you tell us how you came up to think about this drug and start working on it and how the drug works?
So the energy comes from mitochondria. Mitochondria are intracellular organelles that produce ATP utilizing glucose. So mitochondria have their own life within the cell. These are wonderful organelles that really designed to help us to survive and combat stress. But at the same time, mitochondria are the major producers of reactive oxygen species, and oxidative damage associated with this reactive oxygen species production, because mitochondria could go dysfunctional with age, actually contributes to aging and neurodegenerative disorders. So in the lab we have, so I'm a basic scientist, and in the lab we recognized many years ago that mitochondria actually really play a significant role in health and disease. So if mitochondria are dysfunctional, they will contribute to the development of Alzheimer's disease in particular. And we started to think, what can we do to promote mitochondrial health? So our small molecules work in a very interesting way. They go straight through the blood-brain barrier, so our compounds penetrate the brain. They accumulate in mitochondria, and they cause a very mild stress in mitochondria by partially inhibiting mitochondria complex 1, one of the components of electron transport chain involved in the production of ATP. This is very mild. This is almost on the level of metformin, because metformin has very similar mechanisms. But what is happening in response to this mild stress? The cell started to produce more mitochondria, which will replace the old mitochondria that could be dysfunctional and actually renew the mitochondria pool. And that promotes high level of energy and actually reduce oxidative stress. So when we discovered this mechanism, it was stunning, because this is the mechanism that actually is implicated in the positive effect of caloric restriction or exercise. So it is possible that we actually have pharmacological mimetic of caloric restriction and exercise. I'm not saying that you can have this pill and not go to the gym, but it is possible.
What were some of the challenges as a basic scientist, as you said, in transitioning over to becoming a drug discoverer in an academic setting?
For drug discovery, you need a separate PhD, because it has nothing to do with classic chemistry, biological background that we got through our education. The terms, the logic of that, the understanding what does it take to develop the drug is so far away from anything that we learn in academia. And when ADDF, I should say, I wouldn't be talking to you without the support from the ADDF. They were the first to recognize our work as focusing on completely different target for Alzheimer's, because everybody at this time were focusing on amyloid beta. And we said, no, let's focus on mitochondria. It could be even before amyloid beta accumulates, which is true. So we can actually prevent the development of Alzheimer's. So when I was so excited with the small molecule that we basically stumbled over and discovered the mechanism, I came to Howard and said, we have a drug. He said, no, you don't have a drug. I said, no, we have a molecule that works. He said, no, you don't have a drug. And only because ADDF recognized how naive we were in academia, they actually started to put together the seminar series. When people from drug discovery, from companies, from all these regulatory agencies will come and start explaining to us on the level of 101, what does it take to develop the molecule that will go into clinical trials. And I remember my first session, I understood the words, but I couldn't understand the meaning of any of these words. PKPD, ADMET, and they casually go through that, and you sit there thinking, how am I going to navigate? And we did, and it's only because of the support, continuous support of ADDF, education, support. Now we have consultants whom we go to when we need help. We have the network of medicinal chemists, of people who can help with regulatory elements, people who develop the companies and could help to advise what's the best way to bring your compound into clinical trials to avoid any pitfalls when you can lose your compound, because we work so hard to understand the mechanism, to develop the best molecule. So it's incredible, because there is no any other network that could help you to do that as the academic scientist.
If I could ask you one more question, Eugenia. How long have you been working on this?
The first paper came out in 2009, so we count back probably 2005. We recognized this molecule. So what is it? 13?
13 years on the molecule.
On the molecule.
On the drug that you went through. And how long before that were you working on
mitochondria?
The powerhouse of the cell.
Right. So I came to this country in 1994. 96, I started my postdoc at Mayo, and that's basically happened a few years later,
so
in 98.
You've been in this about
20 years.
20 years.
Yeah.
And how close are we to bringing your new drug into the clinic and getting tested in human beings?
We are extremely optimistic, but cautiously optimistic. It takes an army and it takes money, and that's why we're so grateful for your support. Without support of the ATTF and donors and people who are intimately involved in Alzheimer's, who sees Alzheimer's patients. My family is directly affected. I lost a few family members to Alzheimer's. I'm working for myself. I do not want to forget the names of my kids, and for my mother who is still with me, and we're very grateful. But it takes an army, and it's an expensive endeavor. So we are on the path to, we have patents, we have mechanism, and we started to have now a lot of interest and support from the NIH. So we have a big grant to go into mechanistic studies. So I'm very hopeful that we will be there soon.
In the next couple of years.
I hope so.
Okay, thanks.
Thank you.
Okay. Susan, for Catalano, you started a biotech company dedicated to developing treatments for Alzheimer's disease. Could you tell us something about how and why you started a biotech company and what your background is in drug discovery and drug development?
Sure. Before I even get into that, I want to echo what Gia said. Thank all of you. You're just supporting one of the most wonderful organizations out there. It's really making a difference. And
I just want to say that we didn't ask them to start.
It's really, truly true. We are huge fans. The work that you do, Howard, and the staff, it's just wonderful. The Warner family can't say that. So my background is in academic neuroscience, but geez, more than 20 years ago, I went over to industry, to the drug discovery and development industry. And I started this company because after a career in big pharma and medium pharma and startup pharma, I thought, well, you know, I can do a better job. And that's classic entrepreneur. You know, I know how to do it better. So we started from scratch or nothing in 2007. And very early on, the ATTF gave us a very critical grant that really helped us navigate through some tough financial times, which I'm sure you guys remember. I think it was really critical validation of what we've done since then. We've also gotten a lot of support from NIH and have worked closely with our senators from the state of Pennsylvania to really work with Congress to improve the research dollars that are flowing into development, discovery of therapeutics, and advancement of them through the clinic. So we are in Pittsburgh, Pennsylvania, which is a little bit out of the way, but it's been a very crucial part of the success story of the company. There's an awful lot of neuroscience in Pittsburgh, and it's also got about a third of the cost of doing business of anywhere else. So we have a fantastic staff and wonderful people who have been with us as a team since the beginning. We've advanced it from, looking for the drug in the first place all the way through phase 2 clinical trials, which is where we currently are. So we're really excited about what we've discovered and the signals that we're getting back from the clinical trials in Alzheimer's patients.
So can you tell us something about how a drug works and how far along it is at this point?
Yeah, sure. So the drug works by getting rid of the toxin that sits at those synapses that Howard mentioned. These are the points of electrical communication between two brain cells. So the drug is actually going to kick it off and clear it out of the brain. And what you're experiencing in Alzheimer's, what an Alzheimer's patient experiences, is sort of the lights turning off when those points of communication sort of blink out one by one. And you start to have trouble forming new memories, and then eventually it gets worse and worse. And so by getting rid of that toxin, what we've actually seen in our initial studies is that we can turn the lights back on. So the brain's innate capacity to recover from this toxin is pretty remarkable, and the lights actually come back on. So in our initial trials in Alzheimer's patients, these are folks with mild to moderate disease, we actually saw a reduction of these damage markers. So a sign that the damage in the brain is actually being reduced by the drug treatment. So we're very excited to be pursuing that in phase 2 studies. These are studies in about 40 patients that are starting at Yale and other places in Australia and the United States, and should read out in end of next year, beginning of 2020, and we're really excited about that. So again, the support that we've gotten to do this has been a major factor in progressing this far.
We typically think of companies as getting funding not just from government or from philanthropy or venture philanthropy investments like ADDF, but also from investors. And can you tell us something about that ecosystem of how challenging is it to raise money for a small biotech company with an exciting new drug from investors who are risk adverse?
Sure. So I think we've had a little bit of a unique funding history as a company. We are entirely angel funded. So we have individuals as well as organizations of angels. Golden Seeds is one, Tech Host Angels, and other angel groups around the country have been really important for supporting us during this journey. I think these are folks who are impact investors, who have a personal connection to the disease, and are really looking for solutions to the disease. And that's very unusual to be in phase 2 trials and entirely angel backed, not having any institutional investors is a unique profile. We are that's about half of our funding source. The other half is from the federal government and ADDF. So I think that is not an accident. Alzheimer's is really very challenging. I think institutional investors have looked at what has happened in the clinic up until now and really said, this is just too challenging for us. But the one place that, where the one source that really still needs these drugs is the patients and their families. That's not going away. It's not ever going to go away. And I think it's great that in that gap, organizations like ADDF and the federal government have stepped forward and allowed us to proceed. So that's been a big part of our story. And I just couldn't be more grateful to the folks who've really helped us get here.
At what point do you think that a big pharmaceutical company, Lilly, whatever, what are these companies, how would you interface with them? Because we know that, like your phase 2 trials, which are the phase in human testing, regulated by the FDA, where drugs are first tested for their effectiveness and safety, obviously, more safety. But then those trials might be $50 million, for example, which is quite a big sum of money, but maybe angel networks like Golden Seeds and others, as you put together, can afford that. But then when we go to phase 3, which is where drugs have to go in the FDA regulatory process to be proven to have efficacy, those trials cost $300-400 million each, and we have to have 2 of those at least, and sometimes 4. So we're talking somewhere in the range of $1 to $3 billion to actually do the kind of testing that's necessary to get an FDA approval to bring drugs to market for patients. At what point do you see this moving into big pharma, where it pretty much has to go eventually, most of the time, to get an FDA approval?
Absolutely. It's a very good question, and certainly it's one that we ask ourselves all the time. I think there certainly is a role for big pharma in the landscape here. Certainly this is a primary care drug, and as our CEO, Ken Mock, who's sitting right there, likes to say, that's something that's really handled most effectively by these large organizations. So what we've done is, speak with all of them since the company's inception. They've watched us go along. I think as the landscape gets a little bit more risk averse, they move a little bit later, but ultimately they will be, the folks conducting those phase 3 trials, the large registrational trials. I think it's important to also recognize that the agency itself, the FDA itself, is actually really doing a great job responding and dialoguing with the scientists and examining the criteria that it uses to measure the effectiveness of the drug. We've got evidence that our drug is actually really important for restoring brain health. And as they get focused on brain health measures, we think that's a really good thing. So we're really excited about the landscape. Despite the track record that we've had in Alzheimer's disease, we still think there's quite a lot of reason to hope.
And you mentioned the new FDA. Well, the FDA, I think we all would agree, has been very receptive. They also want to see a drug to market for patients with Alzheimer's disease and their families. The FDA people have families that, and they just recently came out with some new guidelines around how drugs might be approved with biomarker evidence. And your company's doing some very unique things with imaging biomarkers in your clinical trials. I wonder if you could tell us, what are biomarkers? Why are they important in drug development? And particularly in your studies, some of the unique things that you're doing with synaptic biomarkers.
Absolutely. So biomarkers are objective measures of change. Usually it's the change in a protein concentration, either in the blood or in the cerebrospinal fluid that bathes the brain and is connected to the brain. These are things that provide an objective measure of change. Up until now, I think what we've been studying as an outcome in these later stage Alzheimer's trials is cognitive performance, or a questionnaire that the patients take that is designed to test how good their memory is and their other kinds of function. And these measures are very fuzzy, they're very changeable. They're very sensitive, and they're not as good as an objective change in the level of a protein in the blood. And it's a little bit like thinking about cardiovascular health. At some point in our clinical trial history with heart failure, we realized that cholesterol levels, which is an objective test that you can do based on a blood draw, were actually really predictive of the outcome of a given therapeutic and the clinical course of the person. And we're just, in the Alzheimer's field, we're just starting to come to grips with that kind of an outcome. And it's a very major step forward, I think, and it's something that we look for. I mentioned earlier that in the trial that we've done in Alzheimer's patients, we saw changes in protein levels of synaptic damage proteins in the fluid that bathes the brain. So these are when the points of electrical communication break down in the disease, they release protein, and this protein winds up getting shipped out of the brain into the cerebrospinal fluid. So when you look in Alzheimer's patients, these damaged proteins are elevated in the cerebrospinal fluid. And so when we looked at Alzheimer's patients after 28 days of treatment with our drug, these proteins, these damaged proteins, were significantly lower. So that's after only 4 weeks. That's not something that's been reported before for any therapeutic. So we're really excited to be treating the patients for longer. And that's something that we're doing in this study at Yale that's enrolling right now. This is a study we're really honored to be doing with Chris Van Dyke and Richard Carson at the Yale ADC and PET Center respectively. They have discovered and optimized a PET tracer that can actually measure the number of synapses, these points of electrical communication in the brain, in a living person. You could never do that before.
PET, you mean an imaging scan?
That's right.
A brain scan, because people might be thinking of dogs now,
It's just, this is what happens when you have a scientist on the podium.
Right.
It's a risk. Yes, this is a PET tracer that's given, so a person is put in a PET imaging instrument. And they're given this tracer, and a picture is taken, and you can see the number of synapses that they have in their brain. And so the trial that we're doing up at Yale is the first trial in the world to use this marker, this measure of the number of synapses in the brain in Alzheimer's patients. And so we're really looking forward to the outcome of that trial. We'll be giving the drug for 6 months and seeing if we can see a slow decline in the loss of these electrical communication points and possibly even an increase.
Thanks. So turning now to prevention, Yuko has been developing our prevention program. Could you tell us something about some of the programs that ADDF is funding in prevention?
Sure. So at ADDF, we have a prevention portfolio, and we're interested in funding studies that address cognitive decline due to aging or due to medical conditions, due to surgery and anesthesia, or traumatic brain injury, as well as menopause-related cognitive symptoms. And I would like to tell you briefly about 2 programs that we're currently funding in this space. So one program is led by Dr. Sharon Inoue at Harvard Medical School and the Hebrew Senior Life Institute for Aging Research. Dr. Inoue is exploring the role of inflammation in long-term cognitive decline following surgery and delirium. Up to 10 million patients experience postoperative delirium each year, and people who have had delirium have a 6-fold increased risk for developing dementia, suggesting that surgery may accelerate the onset of dementia. And so what we currently don't know, so surgery is associated with systemic inflammation, and anti-inflammatory therapeutics might be useful, but what we currently don't know is who is at risk for developing postoperative delirium, because not everyone gets it. Also, we don't know which biomarkers can reliably track inflammation following surgery. So Dr. Inoue is developing a model to predict which patients are at higher risk for developing postoperative delirium as well as cognitive decline. And they are also looking at a lot of different biomarkers that track inflammation, including those in the cerebral spinal fluid and brain scans. So findings from her work will help accelerate development of treatments to counter inflammation related to surgery, while also creating a tool to identify which patients are at high risk. The other program I want to tell you about is a project led by Dr. Christine Yaffe at UCSF. So Dr. Yaffe is exploring the relationship between depression and cognitive decline and dementia. So many studies have suggested the link between depression and dementia risk. However, we don't currently know whether the timing of depression in your life, the timing of treatment, or the types of antidepressants affect dementia risk. And antidepressants have been shown to reduce Alzheimer's markers, biological markers of Alzheimer's disease in preclinical models, like animal models. However, we don't currently know whether antidepressants may decrease risk of dementia in humans. So Dr. Yaffe is performing an epidemiological study pooling data from numerous cohorts that include over 18,000 people, and she's following them. So these people span ages 18 to 90 plus, so she can examine the whole life course. And so findings from her study will help identify if there are any antidepressant treatments that may lower dementia risk in depression.
Thank you. And recently, we've talked a lot about, or we've seen some information about prevention, but there really haven't been any randomized trials. You mentioned epidemiologic observational studies, but there haven't been any really randomized trials of a multifaceted intervention that would incorporate exercise and diet and so on, and to see if we can actually get to the idea of whether or not we can prevent dementia. But recently there's been a couple of studies, that one of which we've been a part of, which is the study in Toulouse, the MAP-T study, about actually seeing if dementia can be prevented in a randomized trial where some people are put into this multifaceted intervention and some aren't. Could you tell us something about those trials?
Sure. So actually last year, the Lancet Commission on Dementia Prevention, Intervention, and Care reported that 35% of dementia cases could be prevented by fully addressing 9 lifestyle factors. These factors include smoking, not completing secondary education, not getting enough exercise, depression, social isolation, midlife hypertension, midlife obesity, type 2 diabetes, and hearing loss. So also the recent trial, so there was a randomized controlled trial called the FINGER trial. So Dr. Fillit mentioned the MAPT trial that looked at the effects of DHA and cognitive training and exercise, and that has shown cognitive benefits. There was a recent trial, the results that came out this year, called the FINGER trial. This was the Finnish geriatric intervention to prevent cognitive impairment and disability, and this took place in Finland. And they found that, this was an interim result that was reported this year, but they found that a multi-domain lifestyle intervention that included a healthy diet, lots of exercise, cognitive training, and management of vascular and metabolic risk factors improved cognitive functions in people with ApoE4, which is the genetic risk factor for Alzheimer's. So what we can say from all of these recent prevention trials is that there are concrete steps you can take to reduce your risk for dementia. And these, include investing in lifelong learning, lifelong relationships and friendships, eating a healthy diet, managing your weight, and if you smoke, you want to quit smoking because smoking does increase your risk for dementia. And if you have conditions such as hypertension, type 2 diabetes, hearing loss, or depression, that you want to seek treatment for these conditions.
So, Dr. Hara, you and your team have written, I think, well over 100 or 150 reports on very specific supplements where you've reviewed all the data from test tubes to mice to human beings on the value of things like CoQ10 and resveratrol and DHA and even reviewed the studies on exercise and so on. And you've published these and presented these on our website, cognitivevitality.org. Could you tell us something more about cognitivevitality.org and its tremendous value for people around the world?
So cognitivevitality.org is a website and a product of ADDF. And so at the Aging and Alzheimer's Prevention Program, we're constantly looking at different foods, drinks, supplements, herbs, natural products, vitamins, as well as drugs. And what we do is we evaluate all of the scientific evidence that's there, because studies in rodents or test tube studies, in vitro studies, they don't really translate that well to potential benefit for humans. So we look at all of the clinical trials if they're available. We also look at all of the rodent studies. We also look at cell culture studies and in vitro studies. And so we rate the level of scientific evidence for that specific compound or drug or herb. For potential brain health benefit, which we also rate, we look at how much evidence there is for that compound and which types of cognitive functions they were able to improve. And some of those studies are in human clinical trials, others are in rodent studies. And most importantly, we also rate safety, because even if something is potentially beneficial, you don't want to ingest something that's really dangerous. And to rate the safety, we look at all of the common side effects, adverse events, severe adverse events, as well as drug interactions.
And I think one of the most important things we try to do is make the evidence and the writing understandable.
Yes.
And credible. And just to add on to what you said, we've cured mice with various supplements and drugs that have been engineered to have the pathology of Alzheimer's disease over 500 times. But as I like to say, mice don't write books, and this is a uniquely human disease, so we really need evidence in humans, and the team's done a fabulous job. And so if you have any questions, Ed, in my practice, in my small practice, sometimes I get people that come in. I had an ambassador once come in who I diagnosed with Alzheimer's, and his wife was at the meeting, and at the end of the meeting, she said, "Can I come see you?" And I said, "Of course." And she said, yeah, and she came to see me, and as she was coming in, she had a carry-on bag, and I thought, well, she's the ambassador's wife, and she's on her way to somewhere in the world. And I said, "Oh, what's in the bag?" And she said, "Well, you told me to empty my shelf in my medicine cabinet and bring everything in." And so I did. And I took a picture of it because very intelligent woman who could afford to see the best doctors in the world, was on about 30 different supplements. And so we lined them up on my desk, and I showed her cognitive vitality, and I printed out some of the reports. And we started throwing everything in the garbage because, especially when you're on multiple medications, then you start having interactions not only among the supplements, but supplements can also affect the medicines that are prescribed by your doctors. So I think we all tend to take supplements, but knowing what the data is behind them is very important. Let me open it up now to any questions that you might have for us, and I'd be happy to. Yes, please.
You mentioned that the biomarkers were more indicative of, a correction of cells. Do you see any time when the FDA might actually use those biomarker tests instead of a very expensive clinical trials? And if so, is there anything we can do to support you with that?
Yeah, so let me take that because our foundation actually supported the first FDA-approved biomarker for Alzheimer's disease. This is a PET scan, so it's a brain scan. And the idea behind it back in 2000 when we started supporting it at Penn was that you could take a molecule that binds the plaques, binds the amyloid. You could radio-label it, right? It could be injected, it would go up into the brain, and if plaques were there, it would bind the plaques and it would light up on a PET scan. And so we supported that program for 4 years at Penn with about $750,000. In 2005, Penn spun out a biotech company called Avid Radiopharmaceuticals with about $7 million in venture capital. And in 2010, that company was bought by Eli Lilly for $800 million. And then in 2012, the company was the first to market with a biomarker and a diagnostic test for Alzheimer's, which I can use in my practice again if I see a patient that has a memory problem. And I'm really not sure what's going on. And it's very early days in terms of their illness. And they want to know. I can just write a prescription. They go down the block to the radiology office. They have that PET scan, which takes about an hour. And within 24 hours, I can tell them with almost 100% certainty whether or not they have Alzheimer's disease. Now, but that's probably not the reason why Lilly bought the test. They're not really in the business of diagnostics. They bought the test because it's a biomarker, and it could accelerate drug development. And that's exactly what happened. And the story there is that at the time Lilly was spending literally, and again, we have to give it to Pharma because they're risking billions of dollars, and over the last 20 years, many tens of billions of dollars on clinical research for Alzheimer's. And so Lilly was doing a clinical trial of an anti-amyloid therapy. And along the way, they had got the test that they bought, and they started testing whether the patients who were being enrolled in this trial, who were being enrolled by dementia experts all over the world, actually had Alzheimer's disease, actually had the amyloid in their brain, which was the target of therapy. And basically up to 35% of the people in the trial had a negative scan. They didn't have amyloid in their brain. They weren't diagnosed properly. And so, of course, you couldn't get a successful trial when 35% of the people in the trial didn't have the disease or the target that you were looking for. But what happened then was that Biogen comes along with another anti-amyloid therapy, and for the first time in the 40 years or so that we've been doing clinical trials, all the people that went into the trial had to have a positive brain scan using the test that we helped to develop. And the brain scan was used over time, over a period of 18 months, to see if the drug worked. Because if the drug worked, then the brain scan would turn negative, right? And that's exactly what happened. Over 18 months, 75% of the plaque in the brain of the people who were getting the therapy at the highest dose, the plaque disappeared. And that was the first time that we really had good evidence in the brain. And the biomarker played a huge role because that was a phase 1B study, what we call just a safety study, with 160 people, 40 in a group. So a very, very small study. But with that, the Biogen skipped phase 2. They went directly to a $3.5 billion program in phase 3 using the test to see if not only did the test remove the amyloid from the brain, but if doing that actually help people to stabilize or get better. And that's where the big numbers come from. So in that case, the biomarker enabled accelerated drug development. And the last thing it did was it enabled prevention trials, because now we can screen people in their 40s and 50s to see if they have Alzheimer's disease. When does it start? And it turns out that it probably starts as early as some people in their 20s, in their 30s, certainly by age 40 or 50. People are accumulating this disease. And so that's enabled prevention trials where we can identify people and give them these anti-amyloid therapies. So the biomarker is playing a critical role today in drug development, just like cholesterol, which was discovered as a risk factor for heart disease in the 1950s. The first drug coming to market to treat cholesterol wasn't until 35 years later when the first statin was approved in 1985. We're just on the cusp of that right now, and it's a very exciting time. And the FDA has recognized that and has incorporated biomarker outcomes as playing an important role in drug approval. Yes. Yeah.
Could you speak to the role of functional medicine and a ketogenic diet and how that would compare to a biomarker in taking a drug?
Yeah, I think ketogenic diet probably up Eugenia's alley right there. So
When neurons stop utilizing glucose properly in the brain, what is happening, the whole system will start now protecting neurons from loss, from degradation. And it's a very interesting process of switching to alternative fuel. So in the cell, mitochondria could utilize few sources of fuel. Glucose would be the one. But if neuron cannot use glucose anymore, by whatever mechanisms and reasons, there is substitution, which the easiest, ketone bodies. Ketone bodies are produced from fatty acids, so it's beta oxidation, and ketones could get straight into mitochondria and substitute for glucose, protecting energy production, because the whole system now is trying to figure out how we can support neuronal function so the brain will function properly. And what is interesting, that part of these fatty acids to produce ketone bodies internally comes from white matter, comes from myelin. And that's why we see reduction of the brain volume because of white matter degeneration. So that is seen on the scans of the human brain, especially in females. It's kind of prominent after menopause. So it's very interesting how brain compensates. So if you cannot fix this problem with glucose utilization, one of the solutions was to substitute the system with ketone bodies. And actually there was a pretty successful trial. I'm not sure was it supported by the ADDF, but I think Alzheimer's Association supported it. Dr. Russ Sverdlov in Kansas did this study when they actually administered ketogenic diet to Alzheimer's patients. It was a short-term trial to see what's happening. And it was stunning because ketone bodies could be measured very accurately in the blood. And what they found, that people with advanced Alzheimer's, so people who could already very well scored on cognitive tests, when ketones were administered, the cognitive tests went up almost in the same parallel as ketone bodies went up. And then they stopped ketogenic diet, and cognitive performance went down almost in parallel to ketone bodies. And he said that it was actually amazing that patients refused to stop ketogenic diet because they felt much better. So it's one of the interesting interventions that probably could work in advanced stages of Alzheimer's disease.
But I just want to temper some of that it's actually kind of difficult, I think, to get on a ketogenic diet. It's like a starvation diet.
Absolutely.
And actually there is a company out Go ahead.
One thing I would like to add to this trial, there were few people dropped out from this trial. And the reason was that it was incredibly difficult for caregivers to provide ketogenic diet. So it actually affects the whole family because you have to switch to completely different style of living and prepare separate food for the patient, and people obviously were not able to manage. So it's really very difficult. Yes, there is a company that is developing ketogenic diet, so might eventually be available as some kind of formulation.
Okay. Next. Yeah.
Hi, can you speak to your family history with respect to if you have someone in your family that has that, or there's a grandparent, several past generations, does that mean that your likelihood of having that gene will be higher, or does it skip generations? Can you speak towards that?
So your question is about the genetics of Alzheimer's disease. And let me say first that people that have mutations in their DNA that cause Alzheimer's disease are pretty rare. It's probably far less than 5% of all the people who get Alzheimer's disease clinically. And these people just have a gene, and I have a patient, for example, that I recently saw who's 49 years old, and she's moderately demented, has tremendous difficulties with language. She got her first clinical symptoms of Alzheimer's at age 45, and her mother got her first clinical symptoms of Alzheimer's disease at age 45. And so this is a rather rare situation. And these people get what we call familial Alzheimer's disease at a very young age. 95% or more of people with Alzheimer's don't have a mutation, but they have a genetic risk factor. And we know a lot about genetic risk factors, genes that are risk factors for Alzheimer's dementia in late life. The most common one we know about is called apolipoprotein E. And this is actually a cholesterol-carrying protein that plays a lot of role in brain repair. So as neurons are constantly being injured every day, we try to repair them. And so the brain makes and the body makes, but the brain in particular makes its own ApoE protein. And there's three kinds. There's ApoE2, ApoE3, and ApoE4. The most common, 70% of us have ApoE3. About 5% of us have ApoE2, and we're protected against Alzheimer's. It's a risk factor. About 20% of people have ApoE4. And people who have a double 4, they get one from their mother, one from their father, have about 15 times the risk of developing Alzheimer's disease. And they'll tend to get it about 10 years earlier. So instead of getting it at 75, they'll get it around 65. And usually there's a family history. And in the clinic, if we see someone who has a strong family history, first of all, just knowing that there's a strong family history increases your risk. But now I can send patients just do a simple blood test, and if they have ApoE4, I know that this is a major risk factor. This is going to contribute to their disease. The thing about it is, and again, I want to bring everything back to how we develop drugs for Alzheimer's, is that we've known about this genetic risk factor for over 25 years. There is no doubt that ApoE4 is a genetic risk factor that if you have a double 4, or even if you have 1 4, you're about 5 times more likely to get Alzheimer's. You're more likely to get Alzheimer's. And 60%, while it's only 20% of the population, 60% of people with an ApoE4 have, 60% of the people with Alzheimer's disease have an ApoE4. So this is a big thing. And you'd think after 25 years, we'd be on the road to developing drugs for Alzheimer's. And we have had an ApoE4 therapeutic portfolio for almost 15 years now. I think we're finally getting it right. We're funding a program at Cornell Medical Center in New York City, where we're taking the gene for ApoE2, and we know from studies that if you're at 2-4, you're protected, you negate the effect of 4. And we're putting that gene for ApoE2 into a virus, a non-infectious virus called an adenovirus. And that virus is injected into the brain. It's gene therapy. It's approved by the FDA. You might have heard in the news today that thalassemia, which is a terrible genetic disease of the blood, can be cured with the same exact kind of gene therapy. Spinal muscular atrophy was just approved by the FDA for gene therapy. The same mechanism, the same model of using this virus to deliver a normal gene to people that don't have the normal gene. And right now we're funding, we've been funding for several years now, a program to take that ApoE4, put it into the viral vector, and that will be given maybe one time to people in their 40s or 50s to prevent Alzheimer's and possibly to cure it in people who have that ApoE4 gene. So I'm excited that finally, after 25, 30 years of knowing about this, we're finally going to be able to drug it and be able to do something about it. And so the other thing I want to say is, again, the difference between biology and developing a drug. different sciences, the difference between genetics and developing a drug. You may hear about a lot of genes that are risk factors for Alzheimer's disease, but getting to do the drug science and make a drug to fix it is a whole nother type of work. Yes.
So I'm Joni Henderson. I'm with the Global Alzheimer's Platform Foundation. We're a partner with ADDF, and I just want to thank Howard and Mark and Waterfountain and our panelists today for all of this great work that you're doing. I wonder, we in this room are very concerned about this disease. We talked about brain health, but what are ways that we who may have issues with our own cognitive functions or loved ones that have cognitive issues? Can the panelists talk about the importance of us getting involved in research and clinical trials, and how important that is to the field?
Sure. Susan, do you want to talk about that?
Yeah, sure. So there are, I think that the recognition is that one of the factors that makes it really difficult to do the clinical trials is actually, getting to the patients, helping them get to the trials, and in recognition of that factor, we have a number of initiatives that ADDF is participating in and the federal government's participating in that are really designed to reach out to the patients, to their families. It's very challenging. Folks with this disease, have disabilities. Their families are overwhelmed. And in contrast to other patient groups that can do a really good job of advocacy for themselves, it's very tough in the case of this disease. So I would encourage everybody to support efforts to reach out to the patients that ADDF and other wonderful organizations are doing. And continue to dialogue with your elected officials, especially in Congress, about the importance of supporting research funding that actually does also support these trials and the outreach efforts.
Yeah, so less than 5% of people with Alzheimer's disease go into clinical trials. It's not that different from some other illnesses, but it's a big problem. Because, as I mentioned earlier, one phase 3 clinical trial that would enroll something like 1,500 or 2,000 people, the cost could be $400 million, but $150 million of those dollars is just trying to find the people for the trials and bring them into the trial. So, for example, we have to screen people. We have to screen maybe 10 or 12 people to find one person who might be eligible for a trial. And if less than 5% of people who are out there with Alzheimer's disease are even going into the screening process, you can imagine how hard it is. So if we funded clinical, we're currently supporting about 25 clinical trials. So we have a lot of experience at the foundation. We have trials that are trying to recruit 50 people to get an early answer in an exploratory phase 2 study, whether a drug even has a signal that it's working. And this recruitment is going on for 5 years. For 5 years, that study at a particular university is trying to find 50 people in 5 years to test this drug. So this is a huge problem that's been raised, and there are increasing efforts to improve recruitment, but it's a very, and again, bringing it back to the perspective, doing this work is really hard. Every step of the way you've heard, the chemistry is hard, the animal studies don't predict what's going to happen in humans, the regulatory environment is certainly improving, and the clinical trials are really hard. Yes. Sorry, I'm not on, let me get on this side because I know I'm not turning right here.
Attention deficit disorders and stimulant therapy, is that related at all to your work? And opiate addiction, are those people going to be more likely to develop Alzheimer's?
Yeah, so I'm going to take that just because, so attention deficit disorder is not a risk factor, but there's a symptom in early Alzheimer's disease that 90% of people have, and it's called apathy. Now, apathy here being used as a neurological term, what it means is that, you see somebody who plays golf and they're retired, and they call their friends and they say, let's go out Tuesday at 4 o'clock or 9 o'clock and the 4 of us will play golf. And then they come into the office and they're not playing golf anymore, but they're really not that demented. Why aren't they playing golf anymore? Why do we see patients in the clinic where the wife will say, well, he was outside riding his bike all the time and doing all these things, and now he just sits on the couch? The reason is a neurological deficit called apathy that occurs in 90% of early stage patients with Alzheimer's. It's a disconnection between motivation and the ability to plan and execute something that you want to do. It's a cognitive deficit. What's interesting is there have been recent studies with essentially Ritalin or methylphenidate that have shown that methylphenidate can improve the symptom of apathy. And what that illustrates not only is that maybe we should be using Ritalin in early stage patients with Alzheimer's disease, and it's safe, but also what we can learn from other diseases for the treatment of Alzheimer's, and that's called repurposing. And the best example of repurposing is thalidomide, which was indicated back in the 1950s for pregnant women with anxiety. Horrible side effects, as we all know. Today, thalidomide is the leading drug on the market for multiple myeloma because it was repurposed. Somebody had an insight that thalidomide might be good for multiple myeloma. It created the company of Celgene, which is one of the leading, most successful biotech companies on the stock market today. So we're repurposing a whole bunch of drugs because the advantage of repurposing is not only time to market. A drug that's in clinical development takes 15 years, $2 billion. But if we test a drug like methylphenidate or drugs like riluzole, which we're conducting a clinical trial, we're funding a clinical trial at Rockefeller University in New York City, a leading drug for ALS, modestly effective for slowing the rate of progression of ALS, slowing it down, rational mechanism of action, could be good for Alzheimer's. So we're studying riluzole, an ALS drug for Alzheimer's disease. And if it works, I can write a prescription, and I'm actually already doing this, for patients off-label, and it would open up a whole new area of research where new chemistry, which is actually already going on, to develop derivatives of riluzole that have new patent protection that will be effective. So as far as opioids, I'm not aware of any evidence, and I don't know if you are, Yuko, as far as opioids. I don't think we've seen any evidence that opioid addiction per se is a risk factor, but I would imagine, quite frankly, that it would be. Because many drugs that affect the brain cause all kinds of disorders, and I think, I don't know that it's actually been studied. I think it's a great question, a very timely question. Yes, please.
I wanted to take a hypothetical person, which could possibly be me, where your mother had Alzheimer's, your father's lucid at 98, and you wonder, am I going to get it or am I not going to get it? Should I be tested or should I just keep my fingers crossed like I do and exercise and eat healthy? You're saying you don't want to know. I don't want to know either. But maybe if they had a drug that could fix it, I would go get tested and I would start taking those drugs.
Exactly.
But right now we don't have that drug, is that correct?
No, but I think the people that ask me, not including myself, if they want to know, they'd say, well, what would I do about it, right? And I was involved in a study that showed that people actually changed their advance directives, they changed their lifestyle, maybe they got more life insurance, whatever, but the value of knowing there comes in with thinking about whether or not, in terms of what you would do about it if you're not symptomatic, about going into research studies, exactly as you said. I think that's a real outcome of being tested. If people got tested in the clinic and then were positive, they'd have more incentive to go into these trials, and that would help with recruitment. And I think we underestimate how important that is to get people in clinical trials.
Hearing loss, this was actually a really new finding that came out last year. And they were results based on an observational study, so it's hard to draw conclusions on cause and effect. But like you suggested, so there may be pathology in the brain that might make people hear less well. But also, if you're not engaged in conversations, or if you're, more reluctant to join, social kind of gatherings, you will probably not get enough cognitive stimulation, and it's probably a downward spiral. So So this is why, I mean, I think the only thing that can be recommended right now is, hearing aids and, just being tested if you have this kind of condition. In terms of cognitive reserve, there's a lot of research on that. So, people who have a cognitively demanding job, or if they have a lifelong cognitive enrichment, they're less likely to get dementia, or if they do, they tend to get it at a later age. So, even if you have risk factors, or even if you have medical conditions, addressing these and, always trying to learn new things, or, taking new classes. What cognitive reserve means is that, even with amyloid beta, or the pathology that's related to Alzheimer's, people who have cognitive reserve are still able to perform at a higher level than those who don't have cognitive reserve. And this is, cognitive reserve can be built by, higher education and learning new things and really keeping your brain very active.
Yeah, and I'll just conclude. Let me just say that it's been known for some time that loss of smell is related to perhaps Alzheimer's disease, and there's a smell test out there that's actually been used. And it may be that we know that hearing loss is actually a neurodegenerative process in old age, and so is loss of smell. So it could be that this neurodegenerative process that affects cognition primarily as a symptom in Alzheimer's is also reflected in hearing loss and in the loss of smell. It could be those are very sensitive peripheral markers. So we don't really know the relationship, but it's there. Well, I want to thank everyone. I invite you to lunch. Thank you for listening.